All right. Very good morning, good afternoon, and good evening, wherever you're from. Thank you for joining our BISMiS Live. Today's is a 3-hour online workshop on 16S rRNA gene sequencing for bacterial identification. As for the speakers today, it's my great pleasure to introduce Dr. Kamlesh Jangid from India. He is a scientist attached to the rRNA Research Institute. He obtained his PhD in 2005. Served at several key positions. He's Associate Chief Editor of Soil Biology and Biochemistry, and he's an editor for PLOS and as well as Frontiers Journal. He is a full member of the ICSP, the International Committee of Systematics and Prokaryotes. He was the immediate president of this society from 2020 to 2023. For his distinguished professional achievements, Dr. Kamlesh was awarded the Honorary Academic Advisor appointments by the China Agricultural University in Beijing in 2018. So Dr. Kamlesh he and his team in ARI is using the conventional microbiology and modern next generation methods or analysis to study microbial life in pure cultures as well as in complex communities. Understanding its ecological role and eventual aim of developing microbial strategies for a sustainable environment. His lab is also deeply committed community science, outreaching, and motivating the younger generation towards scientific research. So without further ado, the 3-hour online workshop on the 16S rRNA genes sequencing for bacterial identification. The floor is yours, Dr. Kamlesh.
Thank you, Aaron. Thank you, Wenjun, and thank you to BISMiS for enabling this platform for dissemination of information. I'm really glad that after hosting nearly for 3.5 years, now I'm able to present something that is dear to me, which is sequencing. As Aaron said, I'm working at Agarkar Research Institute, which is located in Pune, India. And 16S sequencing is something that is still used as one of the key features for identifying bacteria and archaea if there is a potentially novel organism there. So what we thought at BISMiS Live was that we will come up with several workshops that are related to identification of prokaryotes that would involve whether 16S sequencing or whole genome sequencing and also microbiome analysis. So the next 2 workshops will be talking about that. The focus of this workshop will be purely on 16S rRNA gene sequencing for bacterial identification. I would draw your attention to what instructions we have. So I'd suggest you are muted at all times. Please raise your questions in the Q&A box during the theory session so that we can take them at the end of the theory session. We'll take a short 5 to 10 minutes break after the theory session, and we'll come back using the meeting will not be closed, meeting will be on. So we'll take a practical session for about 2 hours after the 10-minute break, where the question answers will be taken on raised hands. It will be easier to answer the questions and queries during the practical session right away. So we will enable your mic during that time so that we can have your questions immediately and get them answered. There has been an email that has been sent to everybody who registered until day before yesterday. And for those who registered since yesterday and early today morning, your Zoom auto message that has been received in your inbox contains the details of the softwares that are required for you to be ready for this practical session. So please, if you have not looked at that email or the Zoom auto message that has come to your inbox, check that link that has come to you for the softwares that needed for your laptops. In the chat box during the question answer session, I'll post a link to download the data, the practice data that we will be using for the practical session. So that link is enabled for download. You can download that data as soon as we finish the theory session. So with these instructions, I would like to begin the presentation about the theory part. The polyphasic species definition for prokaryotes basically identifies an assemblage of isolates originating from a common ancestor population, which is basically defined as a species, and it is typically characterized by a certain degree of phenotypic consistency. That means there has to be a certain number of morphological characteristics and biochemical characteristics that need to be common across these groups, that need to be similar. Then we have the degree of DNA-DNA hybridization, or the similarity between the two DNA, genomic DNAs, should be a significant enough level. And we'll come to that level in the next few minutes. And the organisms must share over 97% of 16S ribosomal RNA gene sequence similarity. So based on this, we define a polyphasic species in prokaryotes. Now, the DDH, or the DNA-DNA hybridization, and the 16S ribosomal RNA similarity, it comes from this graph where you have this species common boundary, which typically is at a 97% cutoff, and anything at a 70% DNA-DNA similarity above that is typically represented by the same species, and anything below that is typically a different, distinct species. This data still holds today in several instances. In fact, majority of instances. There are few where these values are not congruent, especially in certain groups of bacteria. For example, Aeromonas, which is a group where you have a significantly high 16S similarity. Even then, the DDH values are much lower than the 97% cutoff, and because of which there are groups called as genomospecies or phenospecies. So there are certain groups of bacteria where this cutoff doesn't apply. But in general, a 70% DNA-DNA similarity corresponds to a 97% 16S similarity. The contribution of this 97% 16S similarity can be easily visualized or imagined from the fact that when the 16S 97% cutoff was released, the number of prokaryotic species that were described since the 1900s saw an exponential growth in the number of organisms that were, that started to be described per year. And this is based on a data published in 2014 by Jongsik Chun and Fred Rainey in IGSEM. And the number is still continually increasing. Obviously, there are new genomic markers based on which this species definition has kind of shifted a little bit, but it still relies on the 97% cutoff at 16S. The flow sheet for identification of bacteria, basically from a group or a diversity-based study, is you have this isolation of bacterial strains from a niche. You do a DNA profiling or a DNA fingerprinting based on PFGE, RFLP, AFLP, a lot of other DNA fingerprinting methods. to avoid strain duplication. So this is basically done to reduce the number of organisms that you are working with, so to avoid duplication. Then you do a full-length 16S ribosomal RNA gene sequencing of the strains, and you perform a similarity search by blast analysis, or you perform it at various websites that are available, like EasyBioCloud is there. Now, based on the top hits, you construct a phylogenetic tree based on the 16S gene sequences by using at least two algorithms for phylogenetic tree construction. And once you have this, you can once you have this 16S values, you select for strains of the closest strains that have 97% or lower 16S similarity for DNA-DNA hybridization. And you see that if the similarity at DNA-DNA hybridization is less than 70%, if the then if it is less than 70%, you have a new species. And if it is not 70%, it could be the same species. If it is more than 70%, it could be the same species, but then you do a lot of these chemotaxonomic and phenotypic characterization, and you identify the organism as a novel species by following the rules of nomenclature to name the organism, and you submit it in two international culture collections. Recently, there has been an update, a proposal that is floating for this changing in the rules of nomenclature by including the candidatus taxa. So with the new changes, this flowchart is likely to see slight changes. And then you publish this organism in IGSEM or any other recognized journal. The process flow for DNA sequencing is basically you have your target gene amplified by PCR. Now, in our case, this will be a 16S ribosomal RNA gene. The PCR basically comprises of the buffers, the DNTPs, MgCl2, both the primers, the forward and the reverse primer, the template DNA and the tag DNA polymerase that will enable this PCR to happen. Once the PCR is successful, you check it on a gel by gel electrophoresis, and you do a PCR purification of the positive samples. The purification can be done using a spin column or a PEG-NaCl precipitation. The PEG-NaCl is polyethylene glycol and sodium chloride. So basically, it allows fragments beyond a size to be precipitated, and anything that are smaller in size compared to the PEG molecules that you are using or the polymer, it will be remaining in solution, and then they get discarded during the purification step. Followed by this, you perform cycle sequencing, which is basically a PCR but using a single primer, which allows you to, label the fragments with the incorporation of dideoxynucleotide triphosphates into the reaction. And once the cycle sequencing is done, you do a sequencing purification. This can be done again by spin column based or precipitation based, or it could be enzymatic as well. And then you submit them for automated DNA sequencing or conventional Sanger sequencing, which rarely many would do in the lab today because of the affordability of automated sequencing. And then the sequencing will generate a chromatogram that looks something like this during the process of automated sequencing. Now, in this, what you can achieve is you can bifurcate this into two segments. One is the PCR segment, and the other one is the cycling segment or cycle sequencing. We will go into each of these sections one by one so that practically we understand how this thing works. But before we get into the details of sequencing and PCR, we must first understand how does the traditional Sanger sequencing look. So the traditional Sanger's method incorporates radio-labeled dideoxy nucleotides. So you have four separate tubes in which you carry out dideoxy ATP, dideoxy GTP, and dideoxy CTP and dideoxy TTP reactions. And what happens is with the incorporation of the dideoxy nucleotides at the terminal fragments, the synthesis of those fragments is terminated, and then you run these on a polyacrylamide gel as four separate reactions, and you determine the sequence of the fragment starting from the smallest fragment that has traveled the largest distance. So you can do this by interpreting the graph. So the smallest fragment are basically, incorporated here. So you have this, C, G, and so once you run them on the gel, you will have a AT reaction, DG reaction, DC reaction, and DT reaction. And all of these smallest fragments, the last smallest fragment is the C, followed by the G, followed by the A, again A, followed by the C, T, C, G, A, G, T, G, and A. And this is how you start building your sequence as you climb the ladder up. And you will have your sequence from the 5' to the 3' terminal. And you will have this sequence of the fragment as it is generated using the traditional Sanger's method. Now, there is a slight difference in the traditional method and the automated method. In the automated method, the advantage is because the dideoxys are labeled with the fluorescent label dye, you can actually incorporate all the four reactions in a single tube. And the separation of the fragments happens in a capillary electrophoresis. And depending on the fluorescence of the fluorophore, the signal is generated by the base caller and the machine. And the signal is as reproducible as it is in the Sanger method. So that's the advantage. You are not exposing yourself to radioactively labeled dyes, radioactively labeled nucleotides. Instead, you have fluorophores. So you have a better, ease of work and more cost-effective method. So that is how you do the automated DNA sequencing. This is how the automated DNA sequencer looks. So I've given the example of an ABI 3500 XL. So what happens is you basically load your samples onto this plate here. This is a buffer tray, the anode buffer and the cathode buffer. And you have this capillaries, the glass capillaries that are actually running in parallel along with this, electrode. And both of these electrode and the capillary, this tray is moved, and the tray basically allows the movement, and all of these capillaries move into individual wells in the 96 well tray. And through electromagnetic force, the injection happens, and the DNA molecules enter into this capillary, and they traverse this path, this hair-like structure that you see. And over here, there is a solid state laser. There is a laser camera that basically identifies as the DNA molecules, DNA fragments move through this capillary array. You will see a separation of the fragments, and at this position where there is a laser camera, the laser will detect the moving fluorophore, and it will correspondingly give a signal into the computer system. And the kind of signal that is generated is, something like this based on the different fluorophores that are moving here. So this is the kind of machine that is available nowadays. And so that was about the sequencing part. And for the 16S part, it is better to understand first how the 16S molecule basically, allows us the identification of organisms based on conserved regions as well as very hypervariable regions which are spanning across the length of the 16S fragment. So as you can see, there are 9 hypervariable regions within the 16S, which are denoted here by the red blocks. And the blue blocks are the ones which are conserved regions, which allow us to synthesize conserved primers for different regions of amplification. So as you can see, there are multiple primers that are typically, spanning across the length of the fragment, both on the plus strand and the minus strand. And you will have a complete plethora of primers that are typically used. So for the V3, V4 region, which is typically used in microbiome, amplicon-based microbiome analysis, you use this 337 and the 805 primer. And for the V4 region, you use this 553 and the 805 primer. Whereas for the 16S a complete length, you use this 27F or the 8F forward primer and the 1492R primer, which basically binds on the reverse end. So that's how you are able to amplify the entire region using this 27F and 1492R primer. This is a very important slide where you can get the sequences of all the conventionally standard primers that are typically used in the field of 16S sequencing. So these are universal primers plus some sequencing internal primers, which are typically used for sequencing of different regions. For the sequencing strategy, it is first easier to understand how sequencing works. So as we told you, the strategy one is typically using the PCR primers, using the 27F primer. So we know that the sequencing technology cannot amplify the entire 1.5 kb region of the 16S fragment. And because it cannot amplify the entire 1.5 kb region, it is advised to sequence using at least 2 primers. So you have a middle overlapping region where you use the forwards primer from the PCR and the reverse primer from the PCR as your sequencing primers for carrying out the sequencing reactions. And once you get the partial sequencing sequences from the 27F and the 1492 reaction, you can then have an overlapping region with which you can synthesize the full length contig. The other way is to use minimal internal primers. So many people use the 704 forward primer, which basically binds somewhere here, and the 907 reverse primer, which basically binds somewhere here. So that allows the amplification of the reverse orientation. Now, there is a certain advantage of using these internal primers for sequencing. The advantage is that with the PCR primers, at times there is a degenerate amplification at the forward reactions. So because the PCR primer will allow amplification of every fragment which is in the reaction, and if there is a very slight contamination that is happening because of amplification of different operons within the organism, there are at times an organism has multiple ribosomal RNA operons. So that will also co-amplify with the 27F and 1492 reactions. So obviously each of those operons, ribosomal RNA will get amplified. At times you will have a low degree of contamination that is happening, because of which all of the 16S from the contaminant molecules will also co-amplify. And it only happens for the first few reactions, because as the amplification progresses, the target molecules, because of preferential amplification and exponential amplification of the larger quantity of this target molecule, the contaminant will be nullified over a period. But then during sequencing, if you use the 27F primer, the initial signal will be, sometimes mixed peaks, and you will lose some of the signal at the start. And that's why using internal primers is advantageous when you have the two primers only. The best strategy, although, is to use multiple internal primers, where you have the 704F, you have the 536F, you have the 907 R, or the 518R. So these are the different primers which we typically use to ensure that each of the bases within the 1.5 kb fragment is at least read twice. That means you have a 2x coverage for every base, so that you are sure of the correctness of the base call. So that allows one to synthesize or deduce the sequence with higher confidence. Once you have the sequence, this is the sequence analysis workflow that is typically used. You have the raw sequences that are generated by the machine or by your conventional Sanger sequencing method. So you do a manual check of the automated chromatograms that are generated by the system. You ensure that every base call is accurate. Once you are sure of the shorter fragments, you then do a quantic preparation using softwares, or you can do that manually also once the electropherograms or the chromatograms are accurately determined. Followed by that, you do the blast analysis. You can do this on NCBI, and you can also do this on EasyBioCloud. For our demo purpose, we will use NCBI today. Once you have this blast analysis done, you identify the closest relatives of your particular sequence from databases. And once you identify the closest sequences, you build a phylogenetic tree using those closest relative sequences. And based on the tree, the closest clustering organism, you can then get the identity of your organism with an evolutionary perspective, because you have now determined, based on phylogeny, what is the closest relative of your organism. And then you have the eventual identity of your organism. So we'll go one by one through these steps during the practical session. For the theoretical part, what we are showing you here is the raw trace, or the electropherogram, or the chromatogram, as it is typically called in the sequencing field. This is how the electropherogram typically looks. This one is a internal portion, not from the starting of the portion. This is how the system generates this. So we have just used, starting from bases 330 to up till, about 495. So each of this color corresponds to one particular nucleotide call. So as you can see, the blue color is for cytosine, the green color is for adenine, the red color is for thymine, and the black color is for guanine. And the blue bars at the top of this base call indicate the confidence in calling this base, or in determining that this particular peak corresponds to C, this one corresponds to NA, and this one corresponds to NA. So the height and color of this blue bar, it could be a blue bar, it could be an orange or a yellow bar, or it could be a red bar. We will tell you the differences in these base calls as we actually go into the practical session. what does this mean? So these base calls actually represent the quality value or the quality score of the base calling program. Now, once you have this data from the sequencer, it is very important that you separate the data from the noise. And to separate the data from the noise, it is important that you first understand how to read an electropherogram or a chromatogram. As you can see, this looks pretty obvious that there are clear base calls with no, error happening here. This is another example where you see some kind of noise at the base of each of the peaks. Okay, so this noise, although there is a little bit amount of noise, but your actual peaks are very, very clear. They are very clean, and the computer algorithm has no, doubt in ensuring that this particular peak corresponds to a G and this one corresponds to a T, because it has subtracted this noise from the actual data. On the contrary, there are also certain other data sets. As you can see, you will not be able to separate out this, data from the noise. And it is advisable not to use such kind of data because you are going to create artificially parsimonious informative sites into your data set. And by using such kind of data, you are actually going to create artificially new species which in reality do not exist. So as you can see, we only have about 1.5 kilobases or about 1500 bases of the 16S. And out of those, there are only 9 hypervariable regions that typically into the sequence data set, which is only about 1.5 kilobases, you are actually creating artificial noise, which is going to add to novel species when you actually do the analysis at a later, in the later on steps. So manual curation is very, very important. You have to correct for errors which are created because of missed calls. So as I said, when you have mixed peak data, you must throw out this data. This is non-usable data. It doesn't mean anything. At the beginning of the sequence data, you will see that the shorter fragments are very hard to resolve by the sequencing platform. At times, the polymer could be degraded, because of which the system is not able to resolve these shorter fragments. And that's why you have this data which is completely grayed out by the machine. So that clearly indicates that the machine is not confident in calling this basis. You have data which is in red. That means the base score value is below 15. You have this data which is in yellow, which is between 15 and 20. And anything which is above QV 20 or quality value 20 is indicated by the blue data. So I would strongly recommend that whenever you are analyzing data, please use only data sets which have a continuous blue base calls, so that you are confident of that score. Then throughout the read, you will see that the spacing between two base calls is pretty consistent, except at certain places where the system artificially introduces certain base calls because of a shift in the fragment. As you can see, there is no peak in this particular data set, but you have an N incorporated by the system. So one must manually delete this N because there is no corresponding peak for this particular signal. In certain cases, you have this SNP, a real single nucleotide polymorphism that is coming up. And whereas the machine only indicates that there is a C, whereas it should be a SNP that is replaced from a C to a G. It is most likely a heterozygous, allele which is present here, because of which this SNP is occurring. So one must use the data very carefully. One must scan through the chromatogram throughout the read and check each and every position for errors in the data. Near the end of the read, you will notice that the peaks are sort of flattening out. You will see these plateaus and you will not be able to figure out whether there should be, 5 adenine bases here, or there should be 6 adenine bases here, or whether there should be 2 guanine bases, or there should be 1 guanine bases and 1 adenine/guanine base here. So such kind of data set is typically avoided near the end of data. I will tell you the general thumb of rules by which one can edit these data sets. So identifying bad sequence data is very, very important. Whenever you have such crumpy data with mixed peaks, obviously you're not going to use this data set at all. This clearly indicates that there is something wrong with the sequencing reaction. It could possibly mean that there could be multiple priming sites in the sequencing reaction. There could be multiple bands on the gel if you have ran it on the gel before you put it for cycle sequencing. Your PCR product might have generated mixed amplification. Residual PCR primers or primer dimers from the PCR reaction, which could not be completely removed during the purification step. That could be another reason. So incomplete removal of your PCR primers or primer dimers from previous PCR. Your primer may have an high TM, and because of which at low TM it is binding at several positions and it is allowing amplification from several starting points on the 16S fragment or any other fragment in your sequencing reaction. Or it could simply be that the primer has an N-1 population, or a degraded primer is present in your reaction, because of which this could happen. Now, this can happen in several cases. In case you have used the same vial of PCR primers and you have thawed it multiple number of times, continuous freezing and thawing also leads to degradation of primers in a PCR reaction. All right. So sequencing data at the beginning, as I told you already, at the start of the sequencing reaction, you will notice that the peaks are not spaced out properly. They are kind of saturated at the signal. This could happen because of an uneven ratio between the primer to template, which is being used for the cycle sequencing. Your template may be heavily concentrated, or your primer may be heavily concentrated, and it is only being used to generate shorter fragments, because of which saturated peaks can happen. So if it is important for you to determine the start of the sequencing sequencing reads, it is typically advisable that your sequencing primer should bind at least 15 to 20 bases away from your sequence of interest, so that you avoid such issues that appear. Or you can use internal or nested primers for your sequencing reactions. That is also another possibility by which one can do this. As I said, interpreting sequencing data in the middle is also very, very important. Now, imagine this extra space between the letters G and A. All right. There is an extra space compared to the space between other peaks. However, because there is no peak here, the system has not artificially incorporated an additional base. And this is perfectly fine because there is no additional base here. So your sequence is accurate and this can be used. However, in the second case, as you can see, even if there was no peak, the system has added an N, and this N must be manually deleted and it should be corrected for before you use this sequence. And now in this last case, as you can see, there is a very small peak for G, okay, at the bottom. It could be simple background noise, but the system has determined this to be an actual base call and it has incorporated a G here. Now, this can be checked by doing a sequencing for the opposing strand or the complementary strand and confirming whether there is a C on the complementary strand at this position. If there is no C and there is no base call, that means this is an error which has been introduced by the machine, and this needs to be checked for by sequencing the opposing strands. So this needs to be checked for by sequencing the opposing strand. So there are several things that one needs to be sure of before one uses a sequence data. Now, over here, as you can see, the entire sequence is perfectly clean, but here there is a C or there could be a T as well. Now, this could simply mean that there is a single nucleotide polymorphism. In this case, one allele carries a C while the other has a T, and that is absolutely okay. So you should use IUPAC codes for a C/T in this case and check your sequence accordingly. Now, this can happen in case of 16S also, because there are multiple ribosomal RNA operons, the 16S operons in a particular organism. There are several copies of it. And in such cases, you will notice that especially at the hypervariable regions, you will identify several positions which might have a difference in the base at such positions. In this case, in the second case, as I told you earlier, the same example was discussed. The machine has called for a C. If you were to use only the text format or the FASTA format of the sequence, you would not realize that this is actually a SNP where a C/G has appeared. But if you were to check it from the chromatogram, you can carefully check the chromatogram and you can identify that there is a likely C/G SNP that has been called by the machine. So these are several cases where you must ensure that the sequence is accurate and the sequence you use for your analysis is accurate. All right. So at the end, as I told you earlier also, this data seems pretty nice. One can use this data without any apprehensions. However, as I told you earlier, these kind of plateaued peaks do not really give us strong information. One can also make an assumption that by using the base spacing between these bases, one can understand or try to extrapolate or assume that there could be 5 bases here and there could be a G in place of this N here. But that is just an assumption and it cannot be accurately ascertained unless you sequence from the opposing strand. So such scenarios must be avoided. If you have a continuous occurrence of 3 or more Ns in a window of 10 nucleotides, I would stop using that data after that point of occurrences. So we will examine examples during this practical session when we get to it, when we actually analyze sequence data for such scenarios. So it is certainly not advisable to use data set that has multiple N's being called by the base caller. So one should not use such data sets. These are the different scenarios. These are typically called as rolling hills because they have plateaued peaks which are merged between one or the other. So rolling hills are typically not used. So once you have manually curated the data set in the sequencing flow, we saw that contig preparation is one of the key steps. What is contig preparation? Contig preparation is basically when you have these shorter sequences that are generated from the sequencing of a single PCR fragment, it is important to align them and then build a full contig. So you have these separate regions which are coming up, but these overlapping regions that are coming up, you basically align them to obtain a full length sequence. So once you have this full length sequence, this sequence can then be used for your blast analysis or for your other analysis that you need to do. Now, once this full length contig, pictorially, it looks very, very simple. You have this 704F sequence, you have this 536F sequence, and you have this 907R sequence, you have this 518R sequence. Now, these sequences, because 907 and 518, these are the two reverse primers, so they will generate the sequence of the complementary strand. And to align them, you must first reverse complement both of these sequences and do the alignment. So when you do the reverse complement of both of these strands, you will get the forward sequence of this strand and you get the forward sequence of the segment for this portion. And then you can align these together and identify overlapping regions and you can build the full contig. Once you have the full contig, the next step is to do the blast analysis. We will go over how the blast analysis is carried out. You basically input this entire sequence that is generated in the FASTA format into the blast window, and you can use certain parameters. Once you have these blast output, you see that the blast output is typically given, with a lot of hits that come up. Now, as you can see, there is a Bacillus valezensis, there is a Bacillus subtilis, and then there is a Bacillus siamensis. These are multiple strains, but if you see, they all have a very high degree of similarity, 99.93, 99.71, 99.86, and the query coverage is pretty much 100%. In most of the cases, 99% in two of the cases. Now, the typical notion is to pick up the topmost hit and suggest that my organism is Bacillus valezensis. This is not correct. What one should do instead is one should identify the closest relatives from this entire data set. So I've picked out two valezensis sequences for my query sequence, one Bacillus subtilis sequence, one Bacillus siamensis sequence, another Bacillus subtilis sequence which comes from a different strain. This one was NBRC 13719, this is KCTC 3135. And if you go down into that hit, you will also see that there is a Bacillus amyloliquefaciens, there is a Bacillus vallis mortis, there is also another strain of Bacillus subtilis IM12118. So when you actually compare your sequence with all of these sequences using phylogenetic analysis, you will see that your organism actually is closely related to Bacillus siamensis as compared to amyloliquefaciens, valezensis, vallis mortis, or other strains of subtilis. There is also Bacillus inequosorum, Bacillus nakamuri, which have been used here. As you can see, your strain closely aligns with siamensis. And if you were to go with this particular, output, you would have chosen Bacillus valezensis as your identity of the organism. However, it turns out that it is closer to Bacillus siamensis rather than Bacillus valezensis. So this is the beauty of phylogenetic analysis. It tells you how closely related your organism is to the other sequences which are included in the database or in the analysis. So this is very, very important that you should not blindly trust the output of BLAST and use the topmost hit. Instead, you should use multiple sequences that come out from your BLAST analysis and do a phylogenetic analysis completely separately and use this as the identity of your organism. So your identification result will be organism 1 shows closest evolutionary relationship with Bacillus siamensis strain KCTC 13613 based on 16S rRNA gene phylogeny with 99.93% similarity at the DNA level. And hence, organism 1 is therefore identified as Bacillus siamensis. Now, if this identity was less than 97% or between 97 to 98%, my identity would only be given to the genus level. I would resist myself or restrict myself into giving a species level identity because 16S alone cannot identify the organism to the species level unless the similarity is very, very strong. So this needs to be kept in mind that as you get closer to the 97% similarity, your identification can only be given up to a reliable extent of the genus level and not to the species level. So with this, I'll close the theoretical part and I'll be happy to take the questions and remind me to post the link for the data in the chat box. There's a question from Dr. Kunal Jani. If we remove the ends, will there be a change in the actual sequence that will hamper the correct identification of the bacterium? So Kunal, the answer will depend on whether you see corresponding peaks in the data set. If there is no corresponding peak in the data set or in the chromatogram, and you have confirmed it from the complementary sequence generated from the reverse or using another primer, it is okay to remove the ends. But if there are corresponding peaks and the N is incorporated because the base caller is not able to identify the correct peak, then I would leave that N or at least correct it for the correct IUPAC code for multiple nucleotide, at that position. So I would do that instead of, just removing the N. So always make sure that there is no corresponding N, corresponding peak if there is an N before removing it. So that is there. Kumol says, thank you so much for organizing this informative webinar. Thank you so much. We will be heading towards the practical session in some time. Yilmaz has, I want to identify the bacteria in the internal organs of rainbow trout using 16S sequencing. What challenges might I face during 16S gene amplification? Can you provide information on this? Yeah. So whenever you are amplifying 16S from a community of organisms that may be living in the particular niche or the ecosystem that you are looking at, you actually run into the possibility of amplifying or co-amplifying the host DNA as well. There are certain, 16S primers which also co-amplify the 18S portion from the host DNA, especially the mitochondrial 12S. And that is where the problem might arise. So you might have to ensure that all the sequences that are getting amplified from the DNA, whole genomic DNA prep are actually only belonging to the 16S and not the mitochondrial 12S or the 18S portion of your host. So Natasha has a question. The question is, can we, rather than doing phylogenetic analysis for choosing the closest relative, just check the highest DNA similarity presented on the BLAST search? My answer would be no to this, because if we go back to the presentation, you must have noticed that there are a lot of other parameters that are given numerical values. There is an e-score, there is a query coverage, there is a similarity score. All of these parameters are very, very informative, and you cannot solely rely on just the percentage similarity. I'll give you a scenario. Let's say, for example, you have a portion which is aligning 100% similar, but is only about 250 bases long. Your sequence is 1.5 kilobases long. And if you have a 100% similarity with only a 250 bases long, as against a 98% similarity, or let's say a 99% similarity for an entire 1.5 kb region, which one would you choose? This will be questionable if you choose a blast hit based on only 250 base pair, shorter fragment. So all of these other parameters are also very, very important when you look at the blast results. Why 100% similarity of sequence is not taken for the bacterial strain? So as I told you, there are a lot of parameters that need to be looked at. There is the length of the sequence, the coverage of the query sequence, the e-score, the percentage similarity. So all of these will be required before you make an assumption. And that's why phylogenetic comparison is always recommended before you can actually identify a particular organism. So are there any other molecules other than 16S sequencing? Yes, there are many. So for many organisms, gyrase B is used. For many organisms, the RPOD gene is used. So depending on the taxa for which you are trying to look for a closest relative, 16S may not be an accurate marker, and there are other protein coding housekeeping gene markers which are known for identifying those organisms. So in those cases, one must use those well-established non-16S housekeeping protein coding gene markers as the tool for identifying organisms in those cases. So why the peak of each N in electropherogram varies? I could not understand this question. Why the peak of each nucleotide base in electropherogram varies? Okay, this is based on the principle of sequencing. So with every dideoxy incorporated in the fragment, you will have fragments that are generated from 1 nucleotide base, 2 nucleotide base, 3 nucleotide base, 4 nucleotide base long, and so on and so forth up to 1.5 kilobases long. But when you run them on the sequencing machine, you will have fragments which are separated out by 1 bases apart. And that's why the base call is only for that base for which the corresponding fluorescently labeled nucleotide is appearing. So this has to do with the sequencing chemistry rather than, the other portion. Muhammad Mushahid is asking, sir, what we refer to 16S sequencing, because many papers use metagenome sequencing, but it only covers bacteria. But one of the reviewer of my paper restrict me to use metagenomic sequencing, instead use amplicon sequencing in title, paper, and text. Okay, so whenever you are doing 16S ribosomal RNA amplification from a community, it becomes a community study. It does not become a pure culture study. And in those cases, there are two terms which are commonly used. One of them is amplicon sequencing, where your community analysis is based on the amplification of a particular gene from the community. So that is where you use amplicon sequencing. So for example, if you're using a 16S ribosomal RNA V3, V4 as the region of target, you would use 16S V3, V4 for assigning identity to the sequences which are coming up in your sequence data. So in those cases, because it is based on 16S or a gene amplified by PCR, you should use amplicon sequencing. Whenever you are using a whole genome, that means you are extracting whole genome from the community and you are doing shotgun. My assumption would be that you should use shotgun metagenomics as the correct term. So shotgun metagenomics and amplicon sequencing are two separate terms which should clearly identify the origin of those sequences and the experimental strategy that you have used for obtaining those sequences. This identification process is also possible to protists. Yes, the only difference in protists, you should use the 18S counterpart instead of the 16S. Why we don't go directly on phylogenetic results before going to BLAST? Okay, without BLAST, you will not be able to know which of the closest relatives or which of the genus your organism belongs to, right? So to at least know the genus-based identity or the closest relatives, it is advisable to do blast analysis and then identify sequences and then go for phylogenetic analysis. Unless you already know beforehand that your organism is belonging to one of the possible genera, let's say if you already know that it belongs to the genus Pseudomonas, then you will be required to get all the type sequences of all type strains from the genus Pseudomonas belonging to all the species that are described within Pseudomonas, and then you should do the phylogenetic analysis directly. So that can be done, provided you have a priori information about what your organism is. So Eddie is asking, thanks for sharing. Can you explain in more details that the reason why we need to identify the closest relatives and carry out phylogenetic analysis rather than picking up the rank number one top hit? What in phyllo method fix over the latter? I couldn't understand the second part, but as I explained to you that in NCBI blast, the top hit is not always the closest relative to your organism. That's the first. There was a study long time ago. I'm sure NCBI might have curated the data set after that, wherein the authors had looked at the quality of the NCBI database, especially for the 16S ribosomal RNA, and they had proposed that or they had observed that 1 in 20 sequences, so nearly 5% of the 16S data set contained errors. So it is always advisable that when you look at a data set, you should not look at only the top sequence, but also the other sequences that come up in the data set. How to pick the closest relative among the many? As I said, if there are 100 hits and the 100 hits belong to, let's say, 6 or 7 different species within a genus, you should try to get as many sequences of those 6 different species and pick out those to build the phylogenetic tree in your case. Thank you so much for this informational session. Why is the percent similarity of other species of Bacillus more than that of Siamensis to our query sequence? So as I told you, it has to do with the length of the sequence that was examined. If it is a shorter sequence and you get 100% similarity for, let's say, those 200, 250 bases, it will give you a 100% similarity score. But if the sequence is longer and there are one or two base variations, the sequence similarity score will not be 100%. It will be 99 point, let's say 99, 98, 97. It could be that way. So it really depends on a lot of factors. What to do if the bacteria has 2 or more 16S regions in their DNA? In most cases, you will come across organisms that have more than two 16S regions. Luckily, when you do sequencing, it takes care of those 16S regions, and there is not a very high degree of differences in the 16S operons. So you might come across cases where the multiple operons may be actually identical, or there are cases where up to 3% divergence between the multiple operons has been noted. So in those cases, you should clone individual operons and then sequence the clones separately. Is there any possibility of overlapping peaks that might not be visible? If yes, how can we correct them? I don't know if you are pointing towards overlapping peaks, but one of them is not visible. In that case, that means there is no peak. There will not be a case where exactly overlapping peaks are there. In those cases, the software will put an N. And if you look at the raw data, I will show you how to check for those errors in the chromatogram. And when you look at the raw data, you will be able to figure out what different types of colored peaks are there when a base is being called. What is the significance of using a single primer for PCR amplification in cycle sequencing? It has to do with the sequencing chemistry. If you use multiple primers in a single cycle sequencing reaction, you end up getting fragments of all possible combinations from both the sides. Okay, so you'll have one base pair long fragments that have an A and a T because there are occurring bases. You will have 50 base pair fragments from both the ends with multiple terminating terminal fluorophores. So when you load such samples on the automated sequencer, you will see that the sequences are showing multiple peaks. So you never use multiple sequencing primers. How do labeled DNTP function in cycle sequencing, and how do they differ from regular DNTPs? I think that has to do with the basics of cycle sequencing. I will not go into this. The dideoxy nucleotides and the deoxynucleotides have a difference. The 3' hydroxyl group is removed there. So you have a dideoxy, and that is where the extension happens. And because of which wherever the dideoxy nucleotide is incorporated, there is a termination of the chain, and because of which there is no further extension, which is the basic principle of Sanger sequencing. And read more about Sanger sequencing, and this will be clear to you. Professor Ebru Yilmaz, thank you for answering my question. Greetings from Turkey. Thank you so much, Professor Ebru. Why do the results of blast and phylogenetic analysis vary? I have already explained that. Why do they vary? Karan is asking, when interpreting a 16S chromatogram, how can one distinguish between true sequence variation and the sequencing errors, especially in polymorphic regions of the RNA gene? We'll come to that. We'll discuss the real examples, and I'll answer them live when we get to the practical session. So thank you, Karan, for pointing this out. Snehal is asking, can 16S be a good tool for assessing bacterial diversity of freshwater ecosystem, or metagenomics should be preferred? So it really depends what you are looking at. If you're looking at only the bacterial community in the freshwater ecosystem, obviously 16S is the best marker. But if you're looking at the entire community, instead of using a 16S, an ITS, an 18S region separately for separate groups of organism, it is advisable to do a whole metagenome where you will get sequences for all the regions from all the organisms. So you will have a better picture there as to what other organisms also coexist in the ecosystem. Mrunali is asking, sir, can we use any other analysis tool instead of BLAST? Yes, we can. I will show you Easy Texon, which is also available. We will do that. An anonymous attendee is asking, is there any possibility to use 16S sequencing for virus identification? I don't think so. Viruses carry 16S ribosomal RNA in their system. So for viruses, there are other genes that are typically used, especially the ones that encapsulate the viral nucleic acid in the capsid. So proteins that are around the capsid are typically used for viruses. Thank you for sharing this information. Maybe this is beyond the webinar scope. How do you identify viruses and generate phylogeny for viruses? As I explained, you use a different gene and you can use the same phylogenetic tools to study the phylogeny of viruses. You can also use BLAST tool to identify the closest relative once you have the sequence. So that can be done. Ms. Kumar E is asking, sir, if different species observed through BLAST, how can we choose the samples for phylogenetic analysis? I don't know whether you mean different genus or different species. If you mean different species, we have already looked at the example where you are picking up different species of bacillus. But if you have different genera, I would recommend picking different genera as well, sequences from different species of those genera, and then build up the phylogenetic tree. In that case, if you are getting a hit of multiple genera in the BLAST analysis, that clearly indicates that probably your sequence is either very divergent from the known sequences, that means it is likely to represent a novel organism, or it could also mean that your curation of the sequence may be erroneous. That means you have not correctly or accurately corrected the errors in your sequence, and your sequence may actually be a poor quality sequence. So there could be both the possibilities, and one has to look at both the possibilities by going back to the data set and checking for the accuracy of the base calls, corrections that you have done. And if you are confident of that, then look at the possibility that your organism could actually be a novel organism. Sadag is asking which software is good for constructing phylogenetic tree. There are many softwares, but we rely on Mega, and that's what we have sent you as the link to download. The next question is, in my phylogenetic tree, there is a branch named as uncultured bacteria. What does it mean? Okay, so that means these are unclassified organisms which could not be assigned to existing, branches within the tree of life. And that's why your organism is unidentified yet. So that only means that there is more work needed to be done to identify those organisms, try to bring them into culture and grow them in the lab, or identify at least the metagenome or the whole genome of that particular organism from the genomic data, and then identify it as a candidate as species, or identify it as a species under the new seed code. So it depends on that. How do we label DD-CTPs in cycle? So the dideoxynucleotides are either already labeled with the fluorophore dye. You get a ready mix from a manufacturer, or for radioactively labeled, you actually label them with P32 and get those dideoxys separately. That's done. Prasanna Wag has just one word, says sequencing. I don't know what you want to know about sequencing. Anonymous attendee has a question. If my bacteria have more than one 16S and those genes have SNP, which one should I choose? You should use both the possibilities. The current BLAST algorithm is capable of taking the IUPAC codes combinations for all the possibilities of SNPs. Is it only Sanger sequencing is used for 16S sequencing? No. There are other sequencing technologies that are capable of doing Sanger of doing 16S sequencing. Sequencing is just basically you have a strand of DNA that you want to sequence. It doesn't matter whether it is 16S, ITS, guidase V, RPOD, whole genome, or a shorter fragment. You can use any technology to sequence 16S. Depending on the technology, you can only get maybe a smaller portion of the 16S or a larger portion of the 16S. Like if you have the modern technologies of PacBio, you can entirely sequence the 16S, 1.5 kb altogether. So Ravi Kiran Jadhav, what is the criteria to select UPGMA neighbor joining maximum likelihood parsimony to construct phylogenetic? We will come to that when we actually do the phylogenetic analysis in the practical session. Mrunali Ankush is asking, are there any limitations to 16S ribosomal sequencing? Yes, many, but it is still the best tool to answer. One of the biggest limitation, as I told you already, is that 16S in certain cases does not correlate to the similarity which is determined either based on DNA hybridization or the whole genome core gene set. So in many cases, 16S does not have the same resolution power or evolutionary information for many of the genera. So 16S may not be a good marker for that. I already gave you the example of several genera in which this is not typically recommended. So instead you use a protein coding gene for those cases. Yeah. With this, we have finished all the questions that were there. Let me just quickly post the link to download the practice data set in the chat box for everybody.